Welcome to all in my another video of class 12th biology chapter 5 principles of inheritance and variation. I am Dr. Kailashwana, PGT biology Kendriya Vidyalaya Alliance Valsara, and today we are going to discuss Mendel's monohybrid cross. So Mendel, who was also called father of genetics because he performed the very first scientific studies on inheritance on the plant Pisum sativum, which is also called garden pea plant, and by performing those genetic experiments like monohybrid crosses and dihybrid crosses, he observed the data and followed the scientific approach and then concluded some results, and he on the basis of those results he gave some principles or laws, and those laws are also called laws of Mendel's laws of inheritance. So first, before studying the monohybrid cross and its results, let us review the technique of cross hybridization or technique of genetic cross which he had followed. We also discussed this technique in our previous video of genetic terminology. So what was the technique? As we all know that pea plant have bisexual flowers. It means those flowers are having both male and female sex organs, stamens and carpels. Male sex organs stamens and female sex organs carpels. So he selected two plants, one as male plant and another one is female plant. So the plant he selected as female plant, he removed the stamens from that plant, and this step was called emasculation. And then he selected another plant which behaved like male plant. He collected pollen from a normal bush. Then he dusted that pollen on the stigma of the female flower or female plant, and then he covered those flowers and put some label on those flowers, and then he waited to mature those flowers into fruits, and then he collected the seeds and again grow them and then observe the results. So this was the technique of performing genetic cross. Now let us discuss what was the results of those genetic crosses. So firstly, he selected some characters to observe. For example, in every genetic cross he have chosen few features. Few features are listed in this table. For example, seed shape, seed color, flower color, pod shape, pod color, position of flowers, and height of stem or height of plant. He selected these 7 features, and then he performed genetic crosses by using the technique we have discussed before, and then he recorded the results. What results he obtained? That in every condition he was able to see only one character in the next generation of that monohybrid cross. For example, when he selected two variants of seed shape, for example rounded seeds and wrinkled seeds, then he was able to see only rounded seeds in the next generation. All plants were containing only rounded seeds, means spherical seeds, no wrinkled seeds, and this surprised him. And then he selected other features also, for example yellow seeds and green seeds, and again the results were the same because only one feature was he was able to observe in the next generation. All plants are having yellow seeds, not green seeds. Then again he performed the same type of cross with purple flowers and white flowers. Then again in the next generation he was only able to see the purple flowers. So by performing these type of crosses, he concluded that okay, some features are dominant over another features. For example, if in these 7 features he observed rounded seeds, yellow seeds, purple flowers, inflated pods, green pods, axial flowers, and tall stems every time. So he labeled those features as dominant and the another feature as recessive. So in this table we can see that for 7 features, 7 characters, there are 2 different traits, and in every pair there is one trait is dominant and another trait is recessive. What is character and what is trait I also discussed in my previous video. of genetic terminology. So this was the result. Then what he done, that in the next generation, which is called first filial generation also, like this we can represent that genetic growth like this by taking examples of seed shape. So firstly he selected parents with contrasting characters, as I have shown you table. For example, in this representation we are seeing that one seed is rounded and another is wrinkled. This represents complete plants, not only seeds. Plants having rounded seed and plants having wrinkled seeds. And here if we can see the term homozygous. What this homozygous means, as we have discussed earlier in our genetic terminology, homozygous means that having same copies of two alleles. It means the P generation or parental generation is having if parental generation is having rounded seeds, then these rounded seeds are homozygous, means they are having both of the alleles for rounded seeds. And in the same way, wrinkled seeds, the plant having wrinkled seeds also have the same type of alleles for wrinkled seeds. And after the genetic cross in the next generation, which is also called F1 generation. Here F1 means first filial generation, F means phylus and 1 means first. So phylus is a Latin word which the meaning of that is son or the progeny. So here we can conclude that in Mendelian monohybrid cross, F1 generation is the first generation after crossing the homozygous parental generation. So here we can see all seeds are spherical, no one is wrinkled. So then he self fertilizes those plants of F1 generation. So he have done nothing, actually he simply let them self pollinate by covering them so that they will not contaminate. And then the next generation, which was called F2 generation. So what is F2 generation? If we let self pollinate F1 generation, then the next generation we get is called F2 generation. In F2 generation, to his surprise, there were 75% rounded seeds and 25% wrinkled seeds. See, this looks very simple, but it is not as simple as that. So if you can imagine that if we are having two plants, one plant with spherical seeds and another plant with wrinkled seeds, and we are having all plants with rounded seeds in the next generation. But what new thing is happening, that if we take two rounded seed plants from F1 generation and cross them, or if we are having one rounded seed plant and let it self pollinate, then we see in the next generation there are 75% plants with rounded seeds and 25% plants with wrinkled seeds. And here is the result. The exact number was 5474 rounded seed plants and 1850 wrinkled seed plants. So because of this result, what this result show, that this feature we can observe, which is also called phenotypic features, as we have discussed. So this, the ratio of phenotype was 3:1 ratio. If we calculate the ratio, then he formulated some things. That if the reappearance of wrinkled seed happen, or the reappearance of recessive character happens in F2 generation, then this feature must be present in F1 generation. That was his first observation. So how can wrinkled seeds feature can be present in F1 generation, because we are only observing rounded seeds? Then he formulated the laws of inheritance, that F1 generation is showing the feature which is dominant, but they are also, these plants are also having the feature which is recessive. So this is how he concluded that that one plant or one organism have two alleles or two factors or two genetic units for each character. And if any plant or animal have two alleles for one feature, then there is a question arises that whether they are same or they are different. If they are same, we call them homozygous. If they are different, we call them heterozygous. So in this example, we see that F1 generation, these rounded seeded plants are heterozygous, and in P generation they are homozygous. Let us see this cross with the genetic symbols. Then it will be clear to us. So in this diagram, we can see this example with symbols. So here capital R represents the spherical seed and small r represents the wrinkle seed. So as we have learned about terminology also, because that we denote genes with alphabetical symbols, and the various forms of genes which are called alleles, we also denote them same alphabets, but we denote dominant allele with uppercase letter and recessive allele with lowercase letter. So in this example we are denoting capital R as rounded seeds or spherical seeds, and small r as wrinkle seeds. And in next is this flow chart. The gamete formation is showing that during gamete formation these alleles separate, so that each gamete can receive one allele, and after fertilization we have the F1 generation, which all are rounded seeded plants, but their genotype is capital R, capital R, small r, means they are hybrid plants. And when again gamete formation happens after when F1 plants self pollinate, then this capital R and small r alleles segregate from each other. So because of that we are getting 25% wrinkle seed plants in F2 generation. So this is the way to represent this monohybrid cross. And the phenotypic ratio in the F2 generation of monohybrid cross was 3:1 ratio, and if you want to calculate genotypic ratio, we also can calculate, which is 1:1. Capital R 1, capital R 2 plants, and small r only 1 plant. So we can represent this cross like this, or we can represent this cross like this also. There are two ways to represent this. We can see in our NCERT book this pattern is followed. This grid is called Punnett square, which is used to calculate probabilities. And here in this diagram we are representing gametes as in circles, so that we can easily identify the gametes. And here the same example is taken with different symbols. Like here we took the symbol R, alphabetical symbol R, and here we took the symbol S. So it doesn't matter which alphabet we are using. What matter is we are using uppercase letter for dominant one and the lowercase letter for the recessive one. So let's move to the next slide. Let's go in some details of this monohybrid cross. So in this monohybrid cross we have seen 3 generations: P generation, F1 generation, and F2 generation. What is P generation? In Mendelian monohybrid cross, P generation is composed of two contrasting characters two contrasting traits of one feature with pure features. Pure means homozygous. They must be homozygous. If we are saying that we are performing a Mendelian monohybrid cross between spherical seeds and wrinkle seeds, then it must be understood that rounded seeds or spherical seeds must be homozygous. If we are saying that okay, we are having a tall plant and a dwarf plant and we are performing a Mendelian monohybrid cross, then it must be understood that plant is homozygous or pure. So here the P generation or parental generation is shown: homozygous rounded seeds, homozygous wrinkle seeds. Homozygous means having same type of alleles like capital R and capital R, and homozygous wrinkles small r. So the phenotype is rounded and the genotype is written as capital R. So during gamete formation, as we have learned in class 11th, during meiosis, when meiosis happens, actually in plants meiosis happens before gamete formation. It happens in spore formation, but yes, before gamete formation meiosis happens, and after that gametes receives only one allele from each plant, and after fertilization we get the F1 generation, which is first filial generation with rounded seeds. But yes, these seeds are hybrid, and because F1 generation is having hybrid character for one feature, that is why we call this monohybrid cross. We will learn dihybrid cross later, in which we will study two features, or we will get F1 generation which is hybrid for two features. So when we let F1 plants self pollinate, we get F2 generation. Here there is 3:1 ratio of phenotype. We are getting 3/4 means 75% plants spherical means rounded, or 1/4 or 25% plants wrinkled. And if you want to calculate the genotypic ratio, we can also calculate that capital R plants are 1/4, and capital R small r plants are 2 out of 4 and small r plants are 1 out of 4. So this is the simple explanation of monohybrid pros, and the we need to remember the results of the F2 generation in the terms of phenotypic ratio and genotypic ratio, which is 3 to 1 phenotypic ratio and 1 to 1 genotypic ratio. And here are the results of all genetic crosses which Mendel have performed. In this table we can see that this data roughly the ratio is close to 3 to 1 ratio. So this is for all for now, and we will discuss Mendel's laws of inheritance in our next video. Thank you so much.